I will request the chairperson of this session. That's our Dr. S.P. Gupta. Please you take the dais there. So I am very grateful to S.P. Gupta because of his very busy schedule. He was there yesterday, and then he took the flight in the very morning and he reached here at 7:30. This is what actually is his dedication towards the profession. I salute doctors for this kind of attitude and dedication towards the profession, and he has made it possible to be with us this morning. Very with us. Thanks a lot, sir. So I will accompany you and we will start proceedings.
Very good morning, ladies and gentlemen. Welcome to second day of SAPI conference. So today the first session is reproduction, neuroimmune endocrinology. So this session we have 6 lead speakers. I think 4 speakers are ready for their presentations. So since already a delay has happened, so without wasting much time, I think we'll go ahead with the presentations. So I request Dr. A.K. Mishra sir, former SAPI chairman, to deliver his lecture.
May I introduce him to the audience? Dr. Misra has a specialization in chemical reproduction and biotechnology and process technology. He has unique experience of working with the State Agriculture University, University of Agriculture Science and Technology, and Veterinary University, Maharashtra, Animal and Fishery Sciences. He has been there in the National Dairy Development Board for roughly 80 years. Then he is fellow of the recipient of the Free Bhattacharya Award. He has been different capacities from Agriculture and Dairy Board. Professor of Production, University, SKUAST-Kashmir, Vice Chancellor of Maharashtra and Fishery Sciences University. I welcome you sir over here to this conference, 31st SAPI conference. And I am personally grateful to you accepted our invitation and you made it possible to be this morning to deliver a special lecture. Please go ahead.
Thank you to Babu for the good introduction. Honorable Chair, Dr. P.S. Gupta, Co-Chair, very senior SAPI delegates, Dr. President SAPI, Dr. Soki, and others, dear ladies and gentlemen. A very warm morning. In this same environment. At the place heaven on earth. My topic is animals who are waiting for the heart. As the composition are basically non-final production. But very often I see when people talk about animals, they talk about ambitious, in a negative way. And they see, the number of animals which we have in this country are the kind of feel they have. That prompted me to prepare this presentation, to project that the animals are very friends. And animals are contributing in a very significant way to the environment. We all know the contribution of animals. You can see all this. We are the largest producer of milk since the last 25 years, contributing nearly 25% of global milk. The sector is growing very prominently. These days everybody is talking. The national contribution is more than third to GDP, but in many states it is half or more than half of the country. But here I would like to highlight the contribution which that is making. The value was 60,000 crore when this was assessed in 1980. And this value is about 1.5 times of X. And it is more than 125 agricultural communities. And now in the days to come, this value is likely to be much more. That's what I do. This is the situation as far as the global warming and climate change is concerned. Our temperature is already 1.5 degrees centigrade warmer than what was the last decade was the warmest. And in the Paris Agreement, it was decided to bring the temperature increase to 1.5 degrees centigrade above industrial levels. And greenhouse gas emission must be before 2025 and decline by 43% by 2030. But is it happening? I was listening to a report on some 10-15 days back of Secretary of UN, and he said that we are set to increase the global temperature by 2.8 degrees centigrade in this century. And we all know the effect of global warming, climate change. And if we have to sustain the life of the earth, there has to be a healthy environment. And animals are contributing significantly in its stability. From 2006 onward, when West this report came, this report, livestock, long shadow. Since then, everybody talks about greenhouse gas emissions of livestock. The report initially said that livestock contributes something like 7.1 gigaton of carbon dioxide equivalent per year. Of course, subsequently, they reduced it to 14.5%, primarily because of change in the land use calculation. And its greenhouse gases, the methane contribute to 44%, and the waste is consumed by carbon dioxide and nitrous oxide are almost equal. 29% nitrous oxide and 27% CO2. If we see global situation in greenhouse gases, China is number 1, USA is number 2, India is number 3. If you see per capita greenhouse gas emission, equivalent carbon dioxide, Qatar is first, primarily because of gas production. Second is Australia, third is Canada, fourth is USA, and India is the lowest. Look at the figures, 40.5% and 2.5%. The oil share primarily of mineral is 88% of livestock, and out of this 90% comes from animal fermentation. If you see commodity price, carbon dioxide equivalent production per kg of protein produced, for beef it is 300 kg, for meat of sheep goat it is 165, for milk it is 112, and for cow milk, chicken product and pork it is less than 100 kg. So this is the minimum compared to the beef sounds. If you see South Asia, greenhouse gas emission intensity, it is almost double of the global one, 5.5 kg versus 2.7 kg, because our productivity of animal is very, very low, and also the other productive and reproductive performances are not comparable. But this report, as I said, the contributions to the tune of 18% or 14.5%, they are limited. FAO's own scientists direct emission, it is said livestock contributes 5%. For the transport, it contributes 40%. But the moment we calculate on the life cycle basis, where the emissions which are coming out because of feed production, because of the land use changes, because of processing and transportation of animal product, it goes up to 15.5%. But what about the transport? Transport, in this report, even after the life cycle emissions, there is no emission. And it is believed that the greenhouse gas emissions are much lower as reported as compared to what is reported by FAO. Monitored distinctly, they are also looking the FAO. They have such questions as the report. And they said that emissions are lower. It's still based on different calculations, says that the emissions from the livestock are less than 50%. Bushland and Anaheim, they say, of course, that the emissions are much more, 52%. We don't know what resistance they calculate this. And another important thing to consider is in the transport, the gas which is emitted is carbon dioxide, which has the lifespan of 1,000 years as compared to only 10 years of methane. But whatever methane is emitted, this methane production can be minimized. I think NINP and many other institutes, so nowadays we have a full-fledged program in ICR on methane, and the methane production can be reduced to the tune of 20 to 50% by using various methods. In addition, the greenhouse gases can be reduced if we increase the production of our animals. Production as well as if we improve the reproductive performance in terms of age of puberty, age of calving, less mortality and less morbidity.
7878.
Animals are also playing a very, very important role in soil health. As we all know, 95% of the food which we consume comes through soil. And earlier, our forefathers, they were practicing bioagriculture. But we all know, after the Green Revolution, which has contributed in a very, very significant way to the food security of the country, but it has caused a lot of harm to the soil. It has deteriorated the soil health. If we see today, there is low soil organic carbon, declined good microbes and earthworms, impaired inherent resistance of the crop, and because of that there are more incidences of pests and diseases in the crop. Low fertilizer use efficiency, and these days, more and more fertilizer is being used to increase the productivity of the crop. Consequently, there is increased use of inorganic nitrogen. In the last financial year, 120 million tons of nitrogen fertilizer was used. It is important to understand there are a lot of greenhouse gases emission at the time of production of nitrogenous fertilizers, its use as well as after use. And as I said, the utilization efficiency has gone down when it is present in the soil. A lot of it is leaching into the water, nearby water bodies and the food. Because of this, India has become a global hotspot for nitrogen pollution. And this is causing very serious threat to environment and human health. We all know, I can share my own experience when I was a kid, maybe it was in early 70s, in our village, we were producing food grain for our own consumption in an organic manner. And chemical fertilizer was used only in that crop which was disposed of in the market. Even today, we see a lot of reports from Punjab. The Punjab farmers, they don't consume their own wheat. They sell it in the market. This is very alarming. So there is urgent need to reverse the soil degradation for sustainable agriculture production, for sufficient, safe, nourishing and healthy food to remain healthy. Now this is what animals are. They have two main products, dung and urine. Both improve soil fertility. The beauty of the dung is it has more than 60 species of the bacteria and more than 100 species of the protozoa. It can be used as raw or by making compost or by making bioenhancers. The raw is used wet and dry. I'm not going to detail of it because it is not very common. In the form of compost, it is used by aerobic composting, very common, and anaerobic composting or vermicomposting. These days, when people are talking about organic farming and natural farming, they use variety of products like Panchagavya, Bijam. %40 of this dung is used as fuel. We all know, you ladies, they make cow dung cakes and they are used. And I would like to share a report, I think this is for the June 2022, as much as 190 ton of cow dung cake were exported from Goshala to Kuwait. In the Kuwait, it was observed that when they use this dry cow dung cake, the size of the date as well as the quantity of the date, productivity of the date improves. So that's the kind of potential it is having. Urine is used in a similar way. Now the product, I'm not going to the detail, all these byproduct, compost or the farmyard manure or coming up as a slurry after inorganic decomposition or after vermicomposting, they are different. But basically, by and large, they contain the nutrients, they contain the organic matter, some microbes which fix nitrogen from the air. They also contain hormones like auxins, cytokinins and gibberellins. And farmyard manure, once it is applied in a crop, it brings and feeds diverse life. Bacteria, fungi, insects and the worms. And many insects and worms, they burrow the soil and they aerate it, which improves soil fertility. Not only that, the air channels which are created, it holds air, water and the nutrients inside the soil. And this soil organic matter, it maintains soil structure, water retention. It's very, very important. If 5% farmyard manure is added in the soil, it can improve the water holding capacity by 25%. You can imagine how much beneficial it is in those areas where there is rain-fed agriculture. If there is no rain after some time, maybe the seed germination which has taken place may ruin. But if you have used the organic manure or fertilizer, compost in that field, the water is retained in that field for a longer time, and this can be avoided. This water which percolates into the soil, it can recharge the nearby ponds and lakes, and this also decreases the runoff of soil, fertilizer, and the pesticides which pollute water and water. To support healthy plant growth and prevent pollution of the environment, use of compost is important. Animals for natural farming. I think government has appreciated it and they are taking up this in a very, very big area. Under Bharatiya Prakritik Paddhati program, many states are coming forward. One important thing which I would like to share is everybody says if you go for natural farming, it will decline. Yes, it will decline for some years, but even if it declines, it's okay. The kind of food, the kind of expenditure which you will have on your health will be less. Last year, government gave subsidy on chemical fertilizers to the tune of 2.5 lakh crore. Now this amount can, or more amount can be spent on these kind of activities for natural and organic farming. These are the different various programs government has undertaken, and more areas world over. I'm giving this world statistics, I'm not going to detail of it. More and more areas are being covered under organic farming or natural farming. It is said that the organic manure market in India is growing at the rate of 7% CAGR between 2022 to 2032, and it will be of US $1.3 billion in 2032. Now these animals, what is called methane, which is emitted, can be gainfully utilized for gobar gas production. And I tell you, when I was in NDTB, I used this gobar gas for 20 years on the farm without any problem. So this concept is coming, greenhouse, this gobar gas is used for renewable energy, which is a substitute of fossil fuel. These gobar gas plants are coming up for individual farmers, community, as well as on commercial scale. You might have read recently a gobar gas plant of 4,000 cubic meter capacity for electricity production is installed in Varanasi. A gobar gas plant is installed at Manas Vedi Paranpur for 200 cubic meter capacity for the production of bio-CNG. So this is one of the important way we can minimize the greenhouse gas emission by using it for fuel purpose or the production of electricity or CNG. Animal important contribution which animals are making are as animal power. This animal power is renewable, it saves natural resources, fossil fuel, and prevents gas house gas emission. The draft animal number was 49.7 million. If you see the census of 2012, it declined to 38.7 million in 2019. A 2010 report says that to replace 60 million draft animals which were present in 2010, you require 19 million ton of diesel. So you can imagine the greenhouse gas which this will emit and pollute the environment. And 60% draft animal power used in agriculture even today. Because of variety of reasons, some areas machines cannot go. And country is saving diesel to the tune of 2,15,000 million rupees. At present, if we see the draft animals, they are hardly used for 4 months, 2 months in Kharif season and 2 months in Rabi season, and for the rest of the time they are idle. So these animals can be utilized for the production of energy by using variety of equipments. Recently I was seeing a treadmill which a farmer was using for production of electricity. Animals are also used for, especially the cow dung, for other uses. Seed bark for restoration of forest. Cow dung is also used as building material. Cow dung along with the urine is also used as insect repellent. Animals are also used for bee control. And wild animals are also used for variety of purposes of protecting the environment. Elephants create and source water for other species. Wild animals, they reduce fire by grazing, the areas. Rats are the natural pesticides. Squirrels can help trees take root, and whales for carbon sequestration. It is said that a big whale can sequester carbon to the extent of 33 tons. So once a whale is dead, it takes that much of carbon to the bottom of the sea and protect the environment. So, to conclude, I have 3-4 recommendations. Animals which are generally seen as culprit for greenhouse gas emission, they are really boon for soil health, human health, and environment health. There is need to rapidly shift to natural or organic farming, and there is need to use chemicals optimally. We know there are 50 lakh stray cattle in the country which are damaging crops. And these stray cattle can be gainfully utilized to produce gobar gas, organic manure, and electricity. They should not be seen as a nuisance. During transition to natural or organic farming, economic losses be compensated by the government by giving subsidy for this shift and loss. Here I would like to share that the government gave a subsidy of 2.5 lakh crore on fertilizers in the last financial year. Look at the value which is given for chemical fertilizers. For transparency, the process of natural or organic farming practices be digitized using blockchain technology. This is because at some places people are exploiting customers. They sell any product in the name of organic or naturally grown product, which is not correct. I'll close with this quote: there is enough on earth for everybody's need, but not enough for everybody's greed, of Mahatma Gandhi. In the end, I would like to thank organizers, Dr. Pampuri and his team for giving me this opportunity to share my view on animals. Thank you very much.
Thank you, sir, for your excellent and enlightening lecture. So I request the audience if they have any questions or suggestions to the speaker, you please go ahead. We don't have much time, so maybe a limited 1 or 2 questions to the speaker.
What best usage of dung you see?
This is what I have projected. Dung you can use for gobar gas production. You produce slurry, convert it into the compost, and use it as a manure either alone or after supplementing it with the desired nutrients for a particular field. This is the best use.
So we should not check our animal population, domestic animal population.
Well, you should increase the productivity of the animals. I'm not saying you check or you don't check. Even if you have this much of these many animals, but if you can produce, export it. What all I am saying is, you have a population, and in that population, you have a very large population which is also not productive. Now, at present, the rules which we have, you can't do anything. You can't kill them. So the make best use of them. That is what I recommended. They collect those animals, whatever dung they are giving, make use of dung, the dung for production of gobar gas, or if you don't require gobar gas, you can make bio-CNG, you can produce electricity, and in addition to the dung, you can use them, for treadmill or something, some kind of devices, and generate electricity. Make use of them. Why to keep them idle? So that is the only way to use these animals, and all remaining animals, I will say, that use all available technologies to improve their productivity and health. Why our animals should produce less than 2000 kg milk? Our animals should also produce 10,000, 15,000 liters of milk. There is a lot of demand for milk. We should start exporting milk. If we are privileged to have large number of animals, I think we should try to capitalize on it.
But sir, if we see the production cost of our milk as compared to other developed countries, we are having higher value of this per kg milk production than other countries. Who will be taking our milk in that situation?
Our value is not more.
Yes.
No.
Per kg production cost is high as compared to other countries.
No, per kg production cost is high when your animals are giving only, less than 2000 liters, 500 kg, 600 kg.
Even after the best efforts of our organized farms, we are able to produce about 3000 per lactation in case of buffalo. Those are best buffaloes. In case of cattle, it may be slightly more. In that situation, it is not cost effective, sir. Or who will be taking our milk?
No, we are already exporting milk products. Milk products we are exporting.
Product change करके.
Not raw milk. When I am saying export doesn't mean raw milk. Even dung, the Kuwait has imported dung. They have not imported dung as dung. They have imported dung as cow dung cake. There is no sense importing what?
And sir, another thing that we are already deficient in green powder and dry powder, again that the concentrate, and there is a competition between the grain, about the grains, that the human being and that the animals are competing each other. So if we are not reducing the number of animals, how we will feed all this population?
Let me correct myself if I could not communicate. You reduce the low producing animals, but not the animals which are high producing. Or you convert them into high producing animals. That's number one. Number two, we are producing wheat which is surplus. You know how much farmer is getting benefit out of agriculture? Wheat surplus, paddy surplus, even sugarcane surplus. This is so much of surplus. Why can't we convert, we give this particular piece of land for further production? If we are getting more benefit from the livestock as compared to other crops, we should change. Since ages, our fodder production is in the area of less than 5%.
Around 4%.
Less than 5%. So you increase. If there is a market for your animal product, increase.
We are only
Sir, excuse me, sir. Sir, due to paucity of time, sir, please.
last question. We are only
Yeah, both.
पूरा reason है. I think this is the best lecture.
Yes, sir.
And
Yes,
this lecture, the focus is
one minute.
मेरा तो खत्म होने दो डॉक्टर साहब.
हम बाद में भी बात कर लेंगे, ऐसा नहीं है.
One question. We are only considering production of milk from the animal. We are not. We are now giving importance for production of nuts and urine from them.
That's right.
Second thing, you are only talking about rice and wheat. What will be the effect? What will be the use of straw, paddy straw and wheat straw? And you utilize it. Otherwise, you will burn in the environment.
No, ऐसा वो बात नहीं है.
Sir, even after all surpluses, even after all surpluses, the condition of
Humble request to the house. Humble request to the house. We don't have much time. We can have these discussions afterwards at lunch. That will be much better. Yes, please, sir, please. So it's evident. Your lecture was very clear. It's up to you now which technology you want to use. Whether you go for production of byproducts or you go for any other technologies, say reducing animals or increasing their productivity. It's up to your, your institute and your interest in that. But the thing is that lecture was very clear and you have opportunities.
No, but 50 lakh animals.
You can use these.
50 lakh animals, you are talking of using technology or you are using of talking them gobar?
So, thank you, sir. Thanks a lot.
Thanks a lot, sir, for your enlightening lecture, and it draws so many questions and debate and criticism and appreciation. Really, on personal behalf, I love this lecture. So, word of applause to Dr. So, I think next lecture is by Dr. Taru Sharma. I am not able to see her. She is not here, what I learned. So, I request Professor Ashok Kumar Saab to give the next lecture.
Professor Ashok Kumar, who is our lead lecturer from IIT Kanpur. So, a brief about the speaker. He is Professor in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Served as scientist at London University, Sweden for more than 7 years and is a recipient of many more Tata Innovational Fellowship Award by DBT, Madhav Rao India Memorial Award in Science and Technology, Samsung Gioro International Research Award for Health System Research, International Brain Pool Fellow Korea, Executive Board Member for Federation of Asian Biotechnology, FAOB. So this is very brief about the Professor Ashok Kumar, the son of the soil. I may mention. So it's our honor, sir. You have been with us for last 2 days now. I believe so. So despite that the field of your research is coinciding with the field of our research. I mean the physiologists. But the institute where you are working probably that sometimes makes us to think twice. And we are inviting a speaker from IIT. Physiology wale IIT wale people are here. So that way we are very fortunate over here that we are making this as a multidisciplinary and we try to make collaboration, particularly in the science, the physiological science or the engineering science. Why we have that's why we have this designed the topic in such a way technology driven. So we want that technology which you people are here innovating and we want to use that in our biological sciences. That's why we have invited you and I am really thankful to you, grateful to you that you have received our invitation and you are with us. Thanks a lot, sir.
Thank you very much. At the outset, I would like to express my gratitude to the organizers and particularly Dr. Zur Pampuri Saab actually for inviting me here. So, and also I express my thanks to the chair, Dr. Gupta, for the kind words, and all the audience actually. My introduction is just simply, I am the son of the soil actually. I had all my education from this place, then higher education actually outside actually, and then I work at IIT Kanpur actually. I think that is briefly my topic actually. In fact, Dr. Zur has already simplified my being here, why I am here actually and what's my relevance actually in this area actually. I just chose this topic particularly for the reason actually which I am very close with that actually, that this whole work which I am going to just express here actually maybe 20 minutes or all, so is done by the students actually from this valley, the PhD students. So all the work actually has been conducted actually by the PhD students from Kashmir actually. So that's also actually very, very important for me actually to express. So though this is majorly for human health actually, but this has a relevance actually for the animal as well actually. In fact, we have a couple of projects actually which I am discussing here, so which are being conducted at SKUAST. So that makes a sense actually that how these two things can be interlinked actually in the application point of view actually. So before I actually go to the topic, I just want to briefly mention that the last 10, maybe 15 years actually what we have been doing actually in the area of regenerative medicine actually. This is all what I'm talking about, regenerative medicine actually. We chose a major area actually, that's cardiovascular diseases. And in cardiovascular diseases actually, what is the possibilities of actually at least the three major conditions of cardiovascular diseases actually, what can be done by people like us actually as bioengineers actually. So one of them actually is to have actually the stents actually, or you can have actually the what we call it as the grafts, vascular grafts actually. So we develop actually these vascular grafts like here what we see. This is from the technology by what we call as electrospinning actually, or bioprinting technology. So you develop actually these vascular grafts actually, though these are available, these are being used actually in the clinics actually, but what is the importance or what is the advancement actually of these particular type of grafts is majorly actually they are a tri-layered type of graft actually. The inner layer is majorly providing the strength actually, and the outside and the inside layers actually, if you look onto this as a graft actually, this layer or the outside layer actually is containing some specific molecules actually, which are providing the properties actually which has not been there actually in the existing grafts actually. Particularly just reducing the oxidative stress, that's one condition. Another actually which has been shown as failures of the current grafts actually, they try to overcome that actually. And we use in these gallic acid actually, which is present in the green tea actually. Many of you must have seen it actually, or at least know about that actually. So we incorporate that, and the other property actually which these have is they release oxygen actually. Typically most of the critical conditions actually which are developed actually, they lead to the hypoxic condition actually, and that hypoxia does not allow that particular tissue to grow. That's one of the major issues actually. But last 10 years actually we have been working actually how we can supply actually by our approach actually this type of molecule so that these tissues can have a better angiogenesis and can have a better outcome actually. So briefly, this is not something I will majorly discuss actually. So this is one of the important aspect actually. The second one actually, which is what you see at the lower part, it is briefly as shown it here actually, is related to actually when somebody gets a sort of heart attack actually, and there's a clot formation actually. So immediately you give an enzyme which is called as streptokinase actually. In fact, now this enzyme is available in India actually. India produces a lupin, produces this one actually, or urokinase actually. What we have tried to do is actually we of course it is produced actually by the human cell. Now it's produced by the bacteria, streptokinase. So we actually from the particular cell actually which is producing this urokinase, we take the vesicles actually, because they contain the information actually, but many of you must be aware about actually what we call as exosomes. Those are the vesicular bodies, and they contain the information of the cell. So we take those and we have put them actually on a responsive polymer actually. Responsive polymer is like actually that it is like a solution phase actually at room temperature, but as it goes into the body actually it becomes a gel. So if you incorporate these two things together, so you can localize this, put it at that clot, and it completely dissolves the clot as compared to when you put simply a drug, because the drug is exogenously put actually and it goes actually into the systemic, but here it doesn't go into the systemic manner actually. It just focuses at that actually and immediately at least relieves the patient actually who has got a sort of cardiac arrest at that point of time. So moving ahead on that, it's again as I said actually the three conditions actually. Many of you must be aware about actually what we call as myocardial infarction. So again, when there is a cardiac arrest, so then the muscle of the cardiac muscle actually gets fibrotic actually. So that movement of the muscle actually is somewhat hindered. But if it is repeatedly insulted by, let us say actually the person gets actually a second attack actually, then this becomes much more harder actually. Now the bioengineering approaches are actually to develop what you see this patch here. So this patch is again containing the oxygen actually, and then it is also having this, what I said actually as ascorbic acid, which is the antioxidant. So if you put this patch here, where you get actually this type of fibrotic tissue in the heart, so it gets within 2 weeks actually it gets completely recovery of this. But finally this patch is degradable actually, and it is soluble there actually. So you just only create the condition actually and make the tissue, what you see actually here, that the parameters have come back to the normal. So this is one part of actually in the cardiovascular disease actually what we are focusing on. So this was a program actually which DBT gave us with Institute of Liver and Biliary Sciences actually to just tackle the conditions actually of the acute liver failure conditions actually. So people who have finally only a rescue to go actually for liver transplant actually, what can be done for those patients actually, because liver transplant only less than 5% actually we met in India actually, liver transplantation actually. But in that case, what is the possibility actually? Can we have a sort of alternate for that actually in the liver transplantation actually? So this is a bio-artificial liver support device actually. It's on a smaller scale right now actually, but now it is being upscaled actually, and then also getting into the clinical trial. So it's a 3-stage device, what you see it here actually. So the first stage is simply the first chamber in fact actually is just detoxification actually and plasmapheresis actually, that you remove the cells and then you also remove the toxins, because that is typically what the liver failure patients has. Then it moves actually to the second phase, that is here. But this is basically a liver tissue what we can develop outside actually. So it's giving the same functions actually as the liver inside gives you. So now the treated blood, the plasma which is coming from here is now getting actually treated here. Then you have both metabolic and synthesis function here, and then it is circulated back actually into this circle actually. So it is not a complete, I can say actually, a replacement of liver transplant, but there are two situations actually. One, it can regenerate the liver actually, and in that case actually maybe the patient ultimately doesn't need a liver transplant actually if they are lucky for that actually. Second situation is that actually it can also buy the time for the patient actually till the liver becomes available. So it is bridging the gap actually for liver transplantation actually. Maybe you can keep the patient actually for some years till actually the liver ultimately becomes possible for this patient actually. So then this is the work in fact actually which is being carried out at SKUAST. So that's, I just briefly mentioning about this actually, and this is in collaboration actually with Dr. Mudassir Andrabi and Dr. Mudassir Bashir, both of them are from the biotechnology department at SKUAST actually. So this is for the osteoarthritis actually. So we had worked for a long time actually on osteoarthritis actually to develop a sort of engineering approach actually for osteoarthritis actually. Developing a cartilage is not a problem actually, but once you just have actually this implant actually in the knee, it changes its phenotype actually. It becomes fibrotic actually. The clinicians does want to have a sort of longer study. They do want to see actually that if we can show that actually it remains in the same articular cartilage for a long period of time, then perhaps actually they can translate actually into the human application actually. For that, yes, goat is one of the ideal system actually. So what we are doing it here actually, now we are working on the goat model at SKUAST actually. So this is just a picture of the goats what we are holding it here, and right now it's like 8 months actually. We will wait for a few more months and see actually what is the outcome of this study. Main thing is that actually if we can retain this cartilage for a longer period of time actually, it is phenotype, then the clinicians are ready to take it forward actually for the clinical trial actually. So that's what actually this is the purpose actually. Though osteoarthritis and cartilage formation has been going for a long period of time actually. So this is the other two things actually before I move to my topic actually. This is related to the what we call it as a hemostatic bandages actually. You know, lot of blood loss actually immediately becomes fatal. Can we just stop the blood? Can we stop the bleeding, particularly actually if there is a wound, there is a blood injury, or there is a fatal accident actually. So this particular type of thing actually, what I'm showing it here is a hemostatic bandage. So and then we have also the scaffolding system actually for diabetic foot ulcers actually. Again, in the diabetic foot ulcers actually, it's a very, very critical condition actually to treat diabetic foot ulcers actually. How that can be done actually. So we have approach for that also actually, which is presented actually in this, and in 2 weeks of time actually diabetic foot ulcers is possible to be treated. This one actually is now in the clinical trials actually. So these are also some of these applications actually which the students actually from this place particularly who are working for the PhD programs actually have worked on. So we have the peripheral nerve regeneration also actually in this area. So where we have the peripheral nerve cuts actually, or we have actually the conduits which can be repaired actually by these scaffolding system actually and these approaches actually. Will not go on length to this actually, but I will just move to the topic what we talk about the bone substrates. So let me give a little bit of of course I don't need to discuss about the anatomy of the bone here actually. We understand actually it's very much structurally diverse type of thing actually, but that provides actually the unique property to the bone. Okay, so you have the cortical part actually, which is the external, gives the mechanical property, and then you have the internal part, which is the trabecular actually. But what is important actually for the bone actually is hemostasis actually. Bone formation always takes place actually. You get the bone mineralization done, so that has to have a sort of balance actually, what we call it as bone hemostasis. So this is being typically done actually by two types of cells. One is osteoclast actually, and another is one is osteoblast. Osteoblast is responsible for making the bone, whereas osteoclast is responsible for resorbing the bone actually. So if you have the excess of the bone, you resorb that bone actually. So this is a critical thing actually in even the critical defects actually, that you have to maintain actually this hemostasis actually. And that has been our focus, how to maintain this hemostasis actually in the bone formation actually. So bone injuries can be because of the trauma, fragility, fractures, actually, surgical complications actually, but majorly we are focusing on physiological conditions actually. We do want to have actually where the bone diseases like bone infection or bone cancer actually, bone tuberculosis, which is very critical actually. We are focusing on those type of things actually. So what is present at the moment in availability of the bone grafts actually, which is also being used actually for the animals actually. So this topic I particularly chose here actually to put a relevance actually to the animals as well actually. So the same treatments are being done actually for human, which are being done actually in the animals actually. So the gold standard is still the autograft. You take the part of the bone, iliac crust of the person actually, and then you just replace that part actually in the human actually. Or you take the allograft actually from another person actually, or you take actually the xenograft actually. These are typically what normally is being followed actually in the clinics actually. But synthetic bone substitutes have come. So it has been there actually that we now have actually synthetic bone substitute. You don't have to take a bone from the part of the bone actually and then replace it that actually. So there is many companies are there actually which are majorly based on tricalcium phosphate, hydroxyapatite actually. So a range of these are there. One of them, many of you who are aware actually about bone materials actually are methacrylate based bone substitutes actually. But all of them have some limitations as well actually. Like for example in the case of autograft actually, there is a donor site morbidity. Material availability is not that much actually. For example in the case of allografts, so the possibility of disease transmission is actually, and sometimes there can be also graft rejection actually. Polymer based cements actually, these are inflammatory in reaction actually, and then they also have exothermic setting things actually. So what we are doing or what we are looking at in this case is that how taking into account actually what I discussed about the hemostasis, remodeling, and considering that, how these can be designed actually, what you can see here, three conditions which are normally to be met, that the bone materials, they need to be osteoconductive, they need to be inductive, and they need to be having the some cells or the agents from the cells actually. We want to decrease the dependence on the bone grafts actually. That's the purpose actually. So you combine all of them actually. One part obviously is the material part, second part is the bioactive molecules actually, and third part is the cells actually, or you can say actually the cell components are there actually. So we developed indigenously what we call it as hydroxyapatite actually. Some of you particularly who are working in the dental or who are working in the bone area, they are aware about the hydroxyapatite. So if you mix hydroxyapatite actually with calcium sulfate, actually it's a hemihydrate, you get a paste, and this paste is like just a toothpaste type of thing actually, and you can inject this paste. It remains in that viscosity for about 15 minutes actually, but after that actually it becomes a solid pellet, and it's like a cement. But then this calcium sulfate slowly resorbs, and then the ingrowth of the cells goes into directly. So this is what actually is the principle and how it works actually. But that's a base material actually, but you have to make it bioactive actually by incorporating some agents there. So we do that actually, and this is some of the experiments actually which I go through quickly. In the case of where we have actually like the chemical, you have the vesicles or bioactive molecules, you have sometimes actually strontium bioglass actually, and some drug molecules which are used for different type of conditions like doxorubicin for when bone cancer is there, BMP, bone morphogenetic protein, or you have ZA which is used for osteoporosis, or you have actually the antibiotics like in the case of tuberculosis, bone tuberculosis actually you use rifampicin or isoniazid actually. This is one of the examples actually where we just use this for cranial defect. So this was carried out actually in a rabbit actually, and then we just show when you place this cement on the defect actually, which is initially this size of the defect actually, but when you incorporated actually this material and incorporated some of these agents like BMP or ZA, you can see that we can actually cover completely this defect actually, which normally will not be healed on its own because it's a critical defect actually. It cannot get actually healed on its own, and this is just a histology to show that how it has worked actually, and how the proper bone formation has been seen actually in the time period of typically 2 to 4 months, depending upon the type of animal and depending upon the type size of the defect as well. So infection, as I said, bone infection is very common. Bone infection can happen as such directly at the hospital. Infections can happen actually, implant infections can happen, but how can we be able actually to mitigate actually these infections on one hand, and second, after you just have been able to mitigate the infection actually, but how you can be able actually also to regenerate the bone actually there, because you create a sort of major defect actually when you have a major bone infection actually. We call it osteomyelitis actually in that case. So this is some basic work there. I don't want to go into the detail actually. We typically use rifampicin, as you may be knowing actually. Rifampicin is used for the tuberculosis. So, but this is one of the, at least the drug actually, which has also been taking care actually for other type of infections actually, as aureus type of infection, osteomyelitis actually in the case of bone infections actually. But major thrust for us is for the bone tuberculosis actually, which is a major concern actually. So we optimize this whole for that particular thing actually. So this is a tibia model actually in the rat. So you initially create the infection, then you debridement actually because you have to remove the infection from there actually, and when you remove the infection, you generate a large loss of bone there actually. Now this is not just only controlling the infection here, but you have to also now make the bone. So now you fill up this with actually what I showed you, the methyl hydroxyapatite and calcium sulfate actually. Then you can completely, you have 2-3 groups here actually. The one which is having the rifampicin given locally as compared to what you give it orally, systemic way, you have a better outcome on this. So this tells you actually by micro-CT analysis actually that you will be able to completely recover this infection, as well as also be able actually to generate the bone back actually. This is some of the analysis to give you some idea about bone volume and total volume actually actually. And then histology actually also showing that you have getting a proper bone. Typically, if you're in the mesenchymal trichrome histology, you get the blue color like this, then we understand that collagen is formed in that type of condition. Actually, bone tuberculosis, as many of you are aware, actually, this is becoming a sort of really a very difficult condition, particularly actually if it's a spine tuberculosis. Actually, these people are completely, lying on the bed, and sometimes it is difficult actually to treat them. Giving them a treatment for the bone tuberculosis orally, systemic way, actually, it does not reach to the bone at that site so effectively. So our focus is to just provide the antibiotics at the local site where the tuberculosis, where the infection basically is. So you detect the infection, you just do the surgery, minimally surgery, remove the infection there, and place the material at that point of time along with the antibiotics. Actually, so that's what actually the approach is. I don't need to make you people aware about that. Actually, that we have India is on the tuberculosis map on the top. We have 27% actually people having the tuberculosis, but this is total pulmonary as well as extrapulmonary. But extrapulmonary tuberculosis as well is on the higher side. So this is we are conducting this with Jalma in Agra. Actually, this in the guinea pigs. Actually, this particular experiment is done on the spinal cord actually in the guinea pigs. So this Irfan Koyum, actually, he is completed just the PhD actually on this work. So this is what you see actually the spine where you have the infection actually, and then you, as I said, actually there are 4 or 5 groups actually where you have the material. Just to make you clear actually whether the oral antibiotic given is in some way helpful actually, we just obviously have to have a group of that, but then you have the local also as well actually. But in most of the conditions actually we can see that the local delivery is definitely effective and can recover the patient actually. And I should say actually just in 2 months of time actually complete eradication of the tuberculosis at the spine has been there actually. Whereas actually when we give systemically, because one group is systemic also actually by oral antibiotic actually, we have still not been able actually to recover this rat, recover this animal actually guinea pig. So that's a difference between the delivery of actually the antibiotics. The osteoporosis is another condition actually. So osteoporotic, particularly age-related actually, and in the case of women actually after menopause is a major concern actually. Statistically, if you look at here actually, we have 15.8 million people actually, women typically actually with the osteoporotic condition, 4.2 million men above the age of 50. But then this increases the major concern actually in the osteoporosis is the hip injury. So people with the osteoporotic condition actually can have the hip injury, and then there is a need for the hip replacement actually. But this is you can have a hip replacement actually if you have an osteoporotic condition actually, but then the osteoporosis is still there. You may have rectified actually a right hip, but after some time actually you come up with the left hip as well actually fractured. So this was an approach actually. We did this work actually with the Swedish clinicians actually. So they come up with a thought during the time actually when a person actually with a hip injury and osteoporosis coming to the clinic, at that time actually minimally invasive, can we also treat their left hip so that actually their bone formation actually has regained actually. So that was the purpose actually we actually embarked onto this project with that concept. So this is what actually is now shown. Now it is in the clinical work going on Slovenia actually. Again, Irfan has contributed to this work as well actually. So we developed the rat model actually. We just first do the ovarectomy to make the osteoporotic rats actually, and then there's just through this canal what you can see, it's a very refined actually type of thing actually, but he does the surgeries all actually. So he inserted this material with the osteoporotic agents, typically which is called as alendronic acid or bisphosphonate actually. We typically take orally alendronate actually weekly. Many of you may be aware of that. But in this case, we are using actually a drug which is called as alendronate directly in that and see if we can get the recovery back. So that was this experiment actually. And the purpose, as I mentioned to you, was not just only a question of treatment of their hip where the fracture has taken place, but it was also as a sort of preventive effect to see if on the contralateral hip I can actually put this material at that time when the patient has come to the clinic. So can we prevent actually the further, such type of condition in these patients? Actually, this is the data actually related to that actually that we can have when we have locally added this type of material here, that we can have the recovery in this case. So this is the histology. I will not go into the detail of this, but it's just explaining that the outcome and the result outcome has come as expected actually, and the mechanical formation, mechanical properties of the hip injury has come back to the same thing actually. So I will take the last example actually, that's related to bone cancer. So at one point of time actually, if a patient was coming up with any type of bone cancer actually, then only option was to amputate that part actually. Just if it is in the leg, you have to amputate, hand you have to amputate actually. But now we don't need to actually, at least in those situations actually, where we can be able actually to recover it actually. You just first did the debridement, you try to remove basically the tumor at that particular site. But earlier part it was like if you remove that actually, then the bone becomes weak, and then you will not have any possibility actually of regenerating this amount of bone, which is large part of the bone. Naturally it will not come. But with these materials actually and the technologies, now it's possible actually to have that type of possibility. So this is also actually, in this case we in fact also use the herbal on the top actually for for the periosteal regeneration. So we use the doxorubicin actually in this material actually, and we again generated cancer model actually in the rat by giving actually the cells which are the cancer cells, actually SOS cells, and developed the tumor. You can see it here, tumor inoculation actually, and then you did the debridement, you implanted the material actually, and then looked at actually how it works actually. So obviously actually all the marker studies and all actually I did not show actually. So but finally the bone has formed. You can see also from the histology actually, you can also see from quantitative measurements, which is typically the bone mineral density and alkaline phosphatase assay actually, that you can be able to actually, at least in these defects, recover the conditions actually in this. So but now the point is, maybe I will take 1 or 2 minutes more actually. The point is, of course actually this technology seems very applicable. Ultimately you have to bring it actually to the commercialization actually. The commercialization point of view actually, you need to have some manufacturing techniques actually. 3D printing, as some of you must be aware about it actually. So we now do all this actually, which was earlier done manually. We do all this now actually by printing. So 3D printing is just to, as some of you must be aware about, you have a defect actually, like I just choose a defect here. I take a scan of this, and by CAD, I just print it with the printer. Then the printer will give me the same type of structure developed in the same type of shape, size, whatever the materials or which are you using actually. Like you develop actually this here, this type of structure can be developed actually, but the shape can be as desired actually as you take the scan actually in that form. And it's also like you have to have different type of sizes, different type of shapes actually, which are eventually used for the practical purposes. You need to have actually that type of tool. Manually you cannot do that. So we applied this printing in the tibia as well as in the cranial defect model, where you have the cranial defect actually in the head part, actually tibia is just in the leg. So and just implanted these materials, which are printed materials actually with PTMC type of material actually, and then you just saw that actually it is possible to recover these defects in this, just as a continuation of this here actually. So finally concluding actually what we have at the moment actually, we have the materials which are injectable bone cements. This is what we have actually, the nanohydroxyapatite powder, which can be used for the dental actually, which can be used for the bone defects, pillars actually. We now say as what you call as a bone bank basically actually. So while you have all these materials available actually, you have 3D printed type of materials. If the defect is large, then you have functionalized bone cements. You have bone cements or bone implants actually, which are also actually loaded with antibiotics and all. I did not mention only on this part. This is typically what you call it as magnesium implants actually. As you are aware actually, either it is titanium implant or it is SS implant, which are being used as support in in the bone fractures actually, but those needs to have a second surgery actually because you need to take them out. But magnesium is degradable. You may not need to take it out actually. So you just once implant it, it gives the support, it gives the mechanical property, but you may not need to take it out actually. Within a year or so, it can automatically degrade. You don't need to get into this. Yes, we have transferred some of these. Some of them are in the clinics. The technologies are transferred. So this is just like that. Some collaborators, very much thankful to all of them. Actually, we have, as I mentioned, actually, Dr. Mudassir from here, actually, Dr. Mudassir Andrabi from here, actually, many others, Dr. Sareen actually for liver work, actually, and all the funding agencies actually, which I would like to acknowledge. International groups as well were involved actually in some of the works actually from mostly from the Scandinavia, Japan. We have the collaborations actually in this area, and this is my group. Thank you so much for the attention.
I thank Professor Ashok Kumar for a very informative lecture on their excellent bioengineering research being carried out at IIT Kanpur by him and his team. So a few questions due to cost of time. 1 or 2 questions, please.
Sir, first of all, accept my compliment for this informative and very elucidative lecture. But it was very informative and interesting also. Sir, I would like to just 2 queries. The first query, since you are doing it as an implant, it's a bioactive ceramic, won't you require immunosuppression for this because we are using a foreign body? And the second one, what is the advantage of nano hydroxyapatite over the normal hydroxyapatite? Have you seen that?
Thank you very much actually for this interesting question actually. With respect to the immune reaction actually, yes, we do use sometimes immunosuppressant there actually, but in case of this bone substitute actually, we have not seen the rejection actually of this material. Typically, you may have seen actually hydroxyapatite actually in the dental cavities and all actually. Those are all synthetic actually. They are being used actually without any limitation actually. We have not seen that much of a problem. Yes, in case actually we deal actually with like bone cancer or actually in the case of spine, we do feel actually maybe there will be some sort of immune reaction. In that case, yes, cyclosporine actually or any other type of immunosuppressant actually we try to use. So the second question was related to the nanohydroxyapatite. Yes, nanohydroxyapatite actually provides actually a large surface area. So as compared to what you have actually a normal type of hydroxyapatite actually. So that means you ultimately need to have actually a high bone formation actually, mineralization actually. So if you have a size which is smaller, then you will allow actually more cells to penetrate actually through the material. That makes actually the overall bone formation actually more effective. Yes, we have even compared actually because we put obviously for the licensing actually we put to the CDCS, so actually they also asked the same question actually. So in India actually right now there is only one product of this particular bone substitute actually, which is coming in fact from Sweden. In fact, I was involved in the same company, but that is not nano cement actually. So the mechanical property, the comparative study what we did was finally related to this defect which you have healed actually by this material, to check the mechanical property. Ultimately, in the case of bone, it is the mechanical property actually. Can you reach to the mechanical property to the same extent actually which normally a natural bone has? So we saw actually the mechanical property actually in our materials when you use them actually in the model was higher actually than if you use simply the normal type of. I hope I answered your question.
Yes.
Thank you, sir.
Thank you, sir, for your most effective therapeutic implications research. I have only one query regarding the temporary relief to the liver transplant. That is what you have already done. May I know that is depending upon the percentage of the liver damage, to what extent it may be applied. And second is that what is the status of the clinical trial of that particular effort?
Thank you for this question again. Actually. Of course, it depends upon actually the damage which is there. Actually. Of course, that's a question actually which normally the clinicians define whether now the patients need the liver transplant actually, because if all other options are not something actually working for that. Actually. Typically, if you have a cirrhotic, somebody has a cirrhotic condition actually, and then it has a acute on chronic liver failure actually, then even we say, liver is one of the most beautiful organ actually from regeneration point of view. Actually, even if you have 25% liver, healthy liver there, one can still survive on that condition actually. But when it gets cirrhotic actually, then in that case, that possibility doesn't remain there actually. So that time only this possibility of liver transplant becomes actually the possibility in that. I did not, the other approach which we are basically trying, in fact, that may go actually much quicker into the clinical trial as compared to this ball device, because that is a direct relevance. Now we make the liver outside the body, so by printing. So and then we implant that actually into the cirrhotic patients actually, and not the patient actually, but the animals actually. So we got a better outcome in that. Offline we have treated the patient, but online it's a long way. I should not comment here actually, and this is something actually which ILBS people further on will be able to do it actually. But we are for the liver transplant, as an option for the liver transplant, we are using two options. One, this device, which is easier in some way actually to apply, but another is surgical actually. But if you don't have an actual liver available from the healthy donor, you can have the synthetic liver which can be connected actually. So that's the possibility actually of even those. But clinical trial directly to the patient, hopefully we expect actually within a year or so actually if ILBS will take it up further.
Thank you.
Thank you, sir. Since the next speaker is not available, Dr. Kajal Shankar Roy, I invite Dr. Mohan Mandal to present his lecture. So I request Dr. Mohan. Yeah, I request Dr. Mohan to restrict his lecture to 10 to 15 minutes due to paucity of time.
Thank you, respected chairman. I'll try to finish it by 10 minutes, so I can do it fast. Respected Madam sir, we met after a long time physically. My respected teachers. At the outset, I want to thank Dr. Pampuri to invite me to give this lecture. So, without wasting much time, I'm coming directly to this topic, to this kisspeptin. This is the molecule to which I have friendship for last 15 years. I'm in love with this biomolecule because this is having so many things to do in animal science, particularly in the adaptation of reproduction. So, when animal reproduction comes in our mind, although we want that 1 cup every year we'll get out of animal, but it is not always possible. It is well understood facts, already you will know. So, that point of time we generally use generic analogs like butyric acetate in the form of receptor, binaries, so many available. So, though these information are not from bovine, in rodent and then human model, we have seen that if we use generic analog, particularly analog, it repeated use, it causes antibody formation, there is some problem in GI tract. So we search for some alternative molecule, that is two types of stimulatory molecule and inhibitory molecule are there. All are RFMI peptide having two these amino acids terminal in. One is stimulatory to generate, that is kisspeptin, on which I'll be talking, I'll be giving glimpse of my experience in the last 15 years on this topic. Another is inhibitory, we are not discussing here today. So, to make it brief, this 54 amino acid, KP54, this is actually full-length kisspeptin. Actually, this kisspeptin was first discovered in 1996, and it was found to be anti-metastatic factor in case of melanoma cancer. Thereafter, after 6 or 7 years later, in rodent model, in rat model, they found that they also have role in reproduction. Thereafter, in first paper in livestock species came up in the year 2010. I also started working in 2009 onwards in this peptide. As we know, it is potentiated of GNRS. It can replace totally the GNRS. Moreover, it controls the GNRS itself in the hypothalamus. So, already we know that through the estrogenic control, it having negative or positive feedback mechanism in the brain, it regulates KIS1 gene. This novel biomolecule, neuropeptide, first we wanted to standardize the dose, how much dose we should use, because we didn't have any information other than rodent model. So different concentration we tried, though later on we again refined it, the final concentration. Different concentration, we found this concentration 1 microgram per kg body weight in case of bovine is enough to elucidate the effect after, in terms of eligibility, after its administration. So in case, one of my collaborators from Egypt, Animal Reproduction Institute, Gaza, Egypt, he also standardized the doses there in case of Egyptian buffaloes, but their dose is a little higher. So then at the beginning we interested how it is being regulated during Easter cycle. Interestingly, we found 3 peaks of kispeptin throughout the Easter cycle in case of variation in the Easter cycle. Then we thought that buying a kit commercially, it is very costly, around 30,000 to do 30 to 35 samples. So we developed, I'm not going in detail, we developed this second antibody coating technique for estimation of kispeptin in our laboratory, having high sensitivity, 0.1 nanogram per ml, and we can estimate up to 256 nanogram per ml of plasma. It is more than enough for conducting further. We also did parallelism just to see, because here the standard we used was human kispeptin, and we used bovine plasma. So whether they are having homology or not, to test that we run this parallelism test, we found it is almost parallel, means we can do estimation of kispeptin using human kit. Thereafter, we started the effect of kispeptin on ovarian activity, whether they can stimulate ovarian follicular growth or not. Herein we re-standardized our dose. We found that 1.3 microgram per kg body weight is enough to elicit the effect. We found that kispeptin administration causes follicular growth. It especially increases the So during pregnancy also, if you see, the concentration increased linearly over the different stages of stress cycle. So later on we found that placenta is one of the sources of kispeptin in case of bovine and caprine particularly. So if we compare with non-pregnant to early pregnant animal, it is also much higher, at least more than 7 times higher than non-pregnant animal. So we can differentiate by the concentration of kispeptin between the non-pregnant and the pregnant one. So it is different stage. I told it is increasing linearly over the period of pregnancy advancement, and it is expression of KIS1 gene. It is in accordance with the concentration of kispeptin. KIS1R also it is increasing over the advancement of pregnancy, but in meat and meat they are not that much significant. So if you see the application of kispeptin in ancestor species, only 80 animals data. Here we are getting around 57% consumption rate. So later on we found that Kisspeptin what are available in the market in commercially that are not exactly what bovine Kisspeptin is. So what we did, we found 76% similarity. Then we sequenced bovine Kisspeptin and we got the amino acid structure. Then we synthesized our bovine specific Kisspeptin that we are using now. It is more potent. We compared also, I've given one graphical presentation that bovine Kisspeptin with the same concentration giving more release of endogenous LH than human Kisspeptin what are available in the market we used to buy. But we also developed like that of respiratory system relaxant to the asthma patient. We also use these spray devices for using this Kisspeptin. It also enhances the endogenous gonadotropin release and doses much less, 50 microgram per animal, which was otherwise through systemic administration it was 250 to 300 depending on the body weight of the animals. And one spray can dispense out around 20 microgram. So it is easier to use also, everybody can use. There is no need to administer systematically. So here we tested the Kisspeptin used through Kisspep, and in case of GNRs also it is much higher in terms of LH release, so it is more efficient. So this is in Black Bengal goats also tried to see the follicular waves, this follicular growth pattern during anesthesia period during summer months. All this picture shows that it is enhancing the follicular growth and followed by ovulation. It is the C information. So one of my students, he will be presenting today Kisspeptin and libido, sexual behavior and semen quality. So I won't speak much, but what we found that it increases libido and sexual behavior in case of bucks, Black Bengal bucks. So it is very much useful. We have found that during the winter months, bucks are not interested to mount the does, not showing so much sexual behavior required for collection of semen. So it gives good result. We are getting collecting semen by administration of Kisspeptin. So future already Dr. Kadal Shankarai is working in NANP, but and C6 is the best Kisspeptin analog available in the market. We have also through structural modification we designed 3 Kisspeptin analogs. We have analyzed also, I'm not presenting here, but this is having big scope to use the Kisspeptin in the field application because the Kisspeptin normal powder that we use are not stable in room temperature. You cannot use in field, you can keep at room. So to make it stability and more powerful, Kisspeptin specific analogs, particularly bovine specific, is very important. And several behavioral aspects other than sexual and libido, what I told, there are emotional bonding, attachment, there's social behavior, feeding behavior, so many behaviors are related with Kisspeptin. So that has to be studied, we can work on that. And several studies have been conducted on effect of exogenous Kisspeptin on embryo production in vitro fertilization. In cattle and buffalo, only the start. We have started with cattle embryo there at India Rakalani. We are getting good result. One student is working on that. So that's all what I wanted to show you through my presentation. I think I am well in time.
Yeah, thank you so much for that. I thank Dr. Mohan for his nice lecture on commendable work on Kisspeptin. 1 or 2 questions, please.
So, repeated use of this SAPI makes the animal refractory after few treatments. Have you observed this?
Yes.
Then it becomes non-effective.
I didn't get
refractory to this treatment.
I didn't get such a result. I'm using repeatedly, sir, for several experiments with the same animals. I didn't get, but overdose is the problem. Ambulatory stress is there, but you have
Irrespective of the dose, because the antibodies are produced in the animal against this KP. Due to this, the animal becomes refractory and is non-responsive in the coming treatments.
I didn't come across this type of literature, even I am not experienced with that type of problem.
We have some observations related to buffalo on this aspect.
Maybe, but I didn't come across to literature also.
Sir, please accept my compliment. It's really an informative session. Sir, as if I am not wrong, आपने पहला एक भी slide में you have seen, I think you have shown that जो किस peptide है उसका relation है estrogen और LH के साथ.
Now this negative feedback mechanism to control it, like that of GNH or GNR is also being controlled like negative and positive feedback mechanism, whether say pre-evolutionary LH starts. Okay, this estrogen only, steroids, not estrogen exactly, steroids are being regulating this process, whether estrogen has to be released, whether GNRs has to be released, whether there will be release of LH and or gonadotropies may be FSH. This is being controlled by negative whenever required and positive where it is required. This is the mechanism.
Yes, sir. So इसका pregnancy में क्या role है? Non-pregnant or pregnant?
I just wanted to compare, as I shown you that during the pregnancy period throughout the gestation, it is increased linearly. So we have to see early pregnancy means we want to confirm whether the animal is pregnant or not. So if it is the anistase or cyclic, if that concentration of this peptide in anistase or cyclic animal and early pregnant animal is different, is higher or lower, is the concentration is significantly different, we found this is different. That's what I just compared.
Okay, sir. Sir, as you have shown that, I think this experimentally you are using this.
Yeah.
Are you using this clinically? इसका result सब कुछ है clinical जैसे animal में हम अगर use करेंगे इसको.
Clinically in the sense we have used in our farms and we are using in the field also.
Sir, like जैसे in tropical region like Kashmir, इसमें हम ज़्यादा देख रहे हैं जो एनिस्ट्रॉम कंडीशन है ये रिलेटेड टू मान लीजिए न्यूट्रिशनल मैनेजमेंट पे. तो इनमें हम इसको अगर यूज़ करेंगे what will be used and
we got in villages in West Bengal where we are using this in our 2 or 3 adopted villages. There also nutrition is not that much bad but not that much good also. But there it is working. If the animal is debilitated, so reproduction you cannot expect. It should be at least at certain standard nutritionally so that you can apply all this.
Thank you.
Thank you, Dr. Mohan, once again. So the next speaker will be Dr. Ashok Sarvama.
We have with us the speaker, Ashok Barbara, for the information of the House. He is a senior scientist in animal physiology, CIRB, HISAR. His research area is endocrinology, physiology, and molecular biology. I welcome Professor Ashok Barbara to this SAPI conference over here. And I also request him to be brief, within the time, and his talk is very much interesting. I could see urine-based pregnancy diagnosis in the dairy animals. Now that's what we want, that technology should come all the way to build that capacity in the animals. Thank you.
Well, thank you, sir. I'm grateful to you for inviting me here on this conference. And as you told, yes, it's a very interesting field. And when we started working on this, it goes back in the year 2008. When we were given this challenge that why don't you work on pregnancy diagnosis? Because it was one of the mandated areas of CIRB. And pregnancy diagnosis, as you see, is a challenge for all veterinarians. And that is why there were so many interest from different funding agencies, and there are opportunities for working on this area till now. So we all know, I think 90% of the people in this auditorium are veterinarians. And we all know why pregnancy diagnosis in animals is important. It is important for farmers, for the researchers that we have certain experiments, for breeders and clinicians. And above all, it is perhaps the most important criteria to improve productivity of our animals. We have a lot of problems of non-productive animals, and if we can know whether our animal is pregnant or not, so we can rebreed the animal at the earliest possible time, and that's why pregnancy diagnosis is so important in farm animals. And traditionally there are methods, rectal palpation, and just a top-up of it was ultrasonography. Immuno techniques available, especially PEG-based. There are commercial kits available which are based on pregnancy-associated glycoproteins, but they have their limitations. Indirect method with certain genes, MX2 and ISG, these are available, but they are not that accurate and they are very costly also. Indirect method was progesterone also, but it has also its limitations. So if any researcher who will be working on this, then they must have an idea what are the changes that are associated with pregnancy. So there are certain genes which undergo changes, or there are certain endocrinological and autocrine changes which can be targeted for designing some sort of pregnancy diagnosis tool. So the basic question comes, the veterinary sciences or the animal research has been so long, but why we don't have a specific pregnancy diagnosis method as on today? And the main reason for this, the nature itself has made the placentation of wine in such a way that no molecule can as such pass on from a mother to the fetal, or it is available in any sort of biofluid. So we have all the connective tissues from maternal side as well as from fetal side. All connective and epithelial tissues, they remain intact, and it is only very diffuse and only very small molecules can pass from one side to the other sides. And this quaternary placentation is a sort of it's a nature has placed it such a way that the mother will not suffer much damage in any sort of eventuality. And that is why it's so difficult to detect any sort of specific molecules which is related to pregnancy. So when we try to research, or when we have some certain targets in our mind, the first thing that should come in our mind is what can be an itinerary of events that happens during pregnancy. So ovulation occurs and then fertilization within few hours. And after that, it is transported to uterine horns up to 4 days, and hatching occurs by day 7. And maternal recognition, it's on 14th to 19th day, and implantation on 22nd day, and it goes up to even 45th day. So if you look at the day the animal is inseminated till day 45, you are not sure whether that fetus will remain or it is genetically viable or not. So all the genetically viable fetus, they continue in the next trimester, and the animal remain pregnant by that time. So if you look at the losses early in the pregnancy, up to 25% early mortality losses, they have been reported in buffaloes as well as cattle, and that is why on day 30th you declare an animal pregnant on the basis of ultrasonographic examination, but the animal on 16th day, it may be found that there are no sign of any abortions, but the animal is not pregnant, and that is the reason why early embryonic mortality is very important in these cases. So if we are targeting any sort of reliable, by reliability I mean say that you have tested it on a particular day and you are sure that your results will hold good on the subsequent test also, then that is more than 40 days only. And that is why when you are declaring an animal pregnant very early, you may not be sure whether you will be correct or not. So our first thing as far as metabolites was concerned, when we started with the blood, and in that the already established is the pregnancy associated glycoproteins. They are widely used, and it was in Russia that they first reported these packs, and subsequently many groups from NDRI and IVRI, they have worked on it. And if you see, there are 33 isomers of PEG. This but 7 and one more which is not named now. These were reported by NDRI people. So there were 33 and they have a sort of what you can say postpartum release effect. So the animal has parturited, but they will be continued to be secreted in all biofluids up to day 100 or even 150. So you will not be sure whether the PEG that you are getting is from the current pregnancy or the previous pregnancy. That's why a lot of research has gone into it, and still they are targeting PEGs so that to make a specific sort of some test based on these PEGs. So that's how they have said this PEG 7 in case of buffaloes. It was particular, and it is different from if you compare it with the cow. So these were done by, and they have also NDRI, Dr. Sudarshan and Dr. Mohanty, they have developed a test for PEG also, and it will be perhaps sort of just similar to a human pregnancy diagnosis kit, but it will be working on blood. So we have seen some of the metabolites in blood, but then the question came, why not urine? Can urine be a source of some proteins? So that's why we collected some urine, and on the first go we ran a very simple SGS page, and we could not, by naked eye we could not find that there are any differences. But still we proceeded with further, and we could see that there are 1,550 proteins that are visible in urine of a cow. This was reported by, for the first time this was reported in the world, and it was reported from crossbred cows in NDRI. And before this it was considered that only 5 kilodaltons or proteins which have molecular weight below 5 kilodalton, they are excreted in urine. That was a turning point, and people started looking at this diagnostic point of view. Can there be some proteins which can give you answer about pregnancy diagnosis? So bioinformatic analysis of these 1550 proteins, they gave certain amount of information, and particularly they were said that out of these particular proteins, they were mostly signaling proteins which were involved in catalytic functions of and blood coagulation. So the routine type of work, they were assigned to these proteins which were secreted in urine. Based on the SGS page, we cut the gel, and then we analyzed what can be the differences in these proteins which were expressed during pregnancy. So a differential in gel electrophoresis were run, and there seemed to be some sort of differences with respect to the day of as the pregnancy progresses. So there was some differences, but it was not conclusive. So it was never conclusive, and till now we have no answers. There are differences, but your database on the proteins which are related to bovine especially, they are not listed in the available databases. So all these proteins, they are hypothetical, and you cannot go with this sort of experiment. So we planned something very, very simple. So we planned an NMR experiment, and NMR gives you information about the structure of the metabolites that are present in a sample. So we planned an experiment. We selected some 50 animals, 50 heifers. All these were cyclic, and we could get some pregnant animals, both multiparous and uniparous, and then some of those animals we kept deliberately as non-pregnant, and we collected on a daily basis almost. We collected urine, and after day 30th, on the basis of ultrasonography, we declared our samples in a retrospective way that the animals were pregnant or non-pregnant. So this was all how we collected those samples. We did very basic proton NMR, and then the standardized procedures were followed. So out of those samples, we could identify at least 23 metabolites which were present in urine and which were influenced by pregnancy. These were not pregnancy specific, but they do have their levels up or down based on whether the animal is pregnant or not. So out of these 23, we further analyzed what can be, how they can be categorized, and then the advanced regression softwares are available. So we distinguished all the metabolites in two groups. So this was pregnant group and this non-pregnant. So it clearly shows that based on NMR, there are certain metabolites which are present in non-pregnant and which differ in case of a non-pregnant animal. So further, on the basis of day zero is the day when we inseminated our animals, and we tracked these samples up to day 42 to 45. So all these animals, they do have certain specific molecules or specific metabolites which were related to or which were influenced by the pregnancy status of those animals. So three-dimensional principal component analysis did further stress these points that on day zero there were common metabolites and which differed as we progressed with the day of pregnancy. We could identify or we analyzed 123 metabolites in urine, and out of these three, we selected a few. Almost 16 of these metabolites we selected, and when we prepared a VIP score as to how these metabolites they are related to pregnancy, then these top you can say 6 metabolites, all these they are related to tyrosine pathway, and they do have their expression which is influenced by pregnancy. So these fumarate, tyrosine, histidine, 5-hydroxy and tyrosine, they are actually, if their level decreases, then their level decrease, indirectly the other metabolites, their level will increase, and that is why we got the idea that probably this pathway is the one which is most significantly affected by pregnancy. And based on these, we further analyzed on the area coverage to see whether can there be any specific metabolite or a single metabolite which can really tell us the pregnancy status. So tyrosine, anthracynate and 5-hydroxytryptophan, these 4 metabolites, they covered up to only 80%. So if you are taking tyrosine as an indicator molecule, then there are chances that up to 82% you can be sure whether the animal is pregnant or not. Now we have certain metabolites, but how to validate these or how to show whether these are actually overexpressed or underexpressed during pregnancy. So we developed we did some bioinformatics, and we could find out that these are the typical pathways which are influenced by these, and these includes these 5 pathways which came out or which we could relate all these metabolites to these 5 pathways. And this is how these are related to each other, and if we can see their physiological significance, that these are increased during pregnancy, and it is well documented that they have there is increased demand of these metabolites during from maternal side during pregnancy, and they are required for fetal growth and development. They are also involved in serotonin pathway, kynureninic acid, and so many other physiologically important functions are assigned to these metabolites. So therefore, we now have something in mind that we can target these type of groups. And tyrosine and tyramine were the two molecules which we studied from a biochemistry point of view, and then we published all these metabolites recently in one of our very good journals, and we could develop a sort of prototype based on these findings. And I'm deliberately not listing the particular metabolites because of certain intellectual property type of thing. But we have developed this biochemical test. It takes only half an hour, and we have targeted 6 metabolites. From these 6 metabolites, we have targeted a particular functional group, and based on that, we developed a biochemical test. You require only 2 ml of urine, and 3 reagents are there. Only thing is you have to heat the reaction mixture for about 20 minutes at 80 to 90 degrees, and then you will be getting a colored reaction. If the animal is pregnant, you get a colored mixture. Otherwise, it's either very light color or no color at all. Another challenge when we disclose this to the funding agency, DBT, as well as ICR, we were asked to validate our results in certain other species. So we went further and we demonstrated just to validate our results in other species, because we were claiming that it will be common for all ruminants. So we could demonstrate it in methone. With methone, the problem was that all the metabolites which are present in cow or buffalo, they are expressed almost 10 times more in case of methone. So we have to go back to the basics and we have to modify it. So now we have changed this one of the reagents we changed, and we diluted another reagent, and that's why we could get similar results as in cow and buffalo with this modified kit. So in conclusion, there are certain metabolites, yes they are, and because they are indirect indication of pregnancy, so there will be limitations. We have to keep in mind. And the reason is these are not pregnancy specific, these are pregnancy influenced. The metabolites that we have targeted, they are related to embryonic growth. So the embryo grows, and that's how we are getting metabolites in excess in the urine. So now we have targeted something around 6 metabolites, but we have been awarded one competitive grant from National Livestock Mission just to take this work further. And here we will be targeting certain biosensors against these metabolites so that we can skip those heating steps. There are certain other metabolites also which we have not seen, but they can be. Only thing is you have to go further deep and you have to visit your basics and how the animal behaves and how these metabolites can be used as a diagnostic test. So that's all.
Thank you, Dr. Ashok. I compliment for your wonderful work on urine-based pregnancy diagnostics kit. So, 1 or 2 questions, please.
Yeah.
Dr. Blara, as you have told, in case of pets, there is carry-forward effect. Is there any carry-forward effect in these metabolites also?
Yes.
Number 2, by which time this will be available to the end users?
So there is carry forward effect in this also, but it is not up to 100 days or 150 days. Up to 40 days, you can get these metabolites from the previous pregnancy, because all these metabolites, they will be cleared from the liver. The liver hepatic system is there. So that takes some time. So up to 40 days, but in case of large animals just like cow and buffaloes, that's not a problem, because before 40 days, no pregnancy occurs actually, practically. So that is one thing, and we have provided this technology to ICR as well as DBT, and they are in discussion with some private players. So probably it will be available soon. We can hope so. Yes, sir.
Sir, can we use this kit for small ruminant?
We will be trying this time. We have not so far. We have not used because we could not get any samples from sheep and goat. The first reaction from sheep and goat people was why we require any pregnancy diagnosis.
So we are really excited. जब हमने देखा कि यूरिन related है,
Yes.
it was very informative. तो दूसरी बात है सर ये what about early embryonic death? कितने टाइम के बाद ये जो मेटाबोलाइट है ये withdraw हो जाएंगे?
I told it's almost 40 days, and that's why we strongly recommend that this will work only in healthy animals. If you have cases of repeat breeding, then you will not be sure whether the metabolites are from the current pregnancy or from the terminated.
Sir, another thing is that one company is there, I think IDEX rapid visual pregnancy test.
Yes.
तो उनका जो kit है, even we can use that kit. उनका sample है black, लेकिन वो जो kit है, जैसे आपने देखा है within 30 minutes we can get result.
Yes.
So their kit also showing that we can get result within 30 minutes. And moreover, their advantage is that that kit we can use for small ruminant also, like
Yes.
in case of large and small ruminant.
Yeah.
Otherwise, in case of large animal, I think rectal palpation is the best technique. Because in field or anywhere, and any technician can do that one. So I think this pregnancy kit in case of urine, it's important in case of this small ruminant. We hope कि आप उनमें थोड़ा सा कीजिए कुछ.
Yes, sir.
That will be very.
Yes, sir. Your first question was IDEX. So IDEX is based on pregnancy-associated glycoproteins,
Yeah.
bag-based. It's an immuno EIA test. So you can perform it in your lab, and you require blood samples for that.
Yeah.
So that is the primary thing, and it is already available, and it's available across species, across ruminants. I must say.
We can use that small ruminant.
Yeah, you can use in small ruminants.
Sir, and this one economically is it visible, sir? This one जो आपका kit है,
Yes, sir.
इसका cost कितना है?
It will cost only 5 rupees per sample. So it says biochemical. I'm just saying.
I think that kit also they are showing that 96 उनका wells आता है.
हाँ.
उसमें भी cost उतना ही almost उतना ही हो जाएगा.
No, उसमें if your large batch lab
is more easier in comparison to क्योंकि वो आपके control में रहते हैं blood collection. कभी भी आप
मैं वही परेशान है। उसमें आपको ब्लड लेना है, इसमें आपको यूरिन लेना है, तो बड़ा अंतर तो वही है।
large patch lab, they give it for any farmer. They can send their sample and they do it. They have a designated lab in Delhi, I think. So they cost 800 rupees per sample. I don't know, you are getting a 96 well plate, but you have to open it all at once. You have to have 96 samples.
Heart, it will be very good for a heart.
Yeah.
State.
Yeah, I think it's time now. I request the audience to pose questions. Will be available. So I think we'll end this session now. It's already time. So
Yes, before we break for tea over here, we'll have to honor our lead speakers over here. May I request Rais, please go for those things. It's again request to all those guests which are leaving the station, please fill up that form so that we can know their itinerary of, let's say, the departure times, so that the arrangements will be made. Please see that they hand over that form to any of our,
Thank you, sir. I would like to call the chairman of the session to come on the stage and receive the memento. First, I think chairman sir will felicitate our lead speakers. I call Dr. A.K. Mishra sir kindly come on the stage to receive the felicitation from the chairman of the session, Dr. P.S. Gupta sir. Light.
लाइट ऑन कर लीजिए, लाइट सारे लीड पेपर Thank you, sir. Sir, Professor Ashok Kumar. I request Dr. Mohan Mandal, sir. Now I request Dr. Ashok Kumar Balhara, sir, to receive the moment.
Congratulations, Dr. Mandal. Thank you, sir. How is our another man? He was with me.
I request Dr. S.K. Rastogi, sir, to felicitate our chairman of the session, Dr. P.S.P. Mittal, sir. I request Dr. J.P. Mittal, sir, to felicitate our...
Sir, please be this.
Sir, receive कर लीजिए, कोई दिक्कत नहीं। आप रिसीव नहीं करेंगे? Thank you so much. We have a tea break and sir, before tea break, I will like to announce one or two announcements are there. One announcement: we have arranged a free health camp outside our auditorium. There are the doctors from the Cygnus Ujala Hospital, which has been recently inaugurated in Kashmir by the LG of the UT. Second announcement is those who have already registered for the SAPI live membership, certificates are there outside where you have yesterday registered yourself. You can receive your certificates from that hall. And after that, we have a group photograph, I think, in this hall. Well, I think right now we will take a group photo of. Otherwise, then. Sir,
we will break for tea.
Okay, sir.
We will break for tea, then we will assemble.
Sir, Ashok sir जा रहे हैं sir, इसलिए. Sir, I think.
Sir, please, सब आ जाइए.
Very quickly, within 2 minutes. Everyone is requested to come on the stage.
जो बाहर भी होंगे आ जाइए, please.
Kindly, please. Friends, sir, light के तो सर यहाँ. Light.
Projector करें बांध.
Projector बांध तो light रही यार. Okay. आत्मा सना वो कोई. आगे सर. आत्मा सना वो कोई बैनर लग गई.
ये reflection.
अच्छा ये पे थोड़ा kindly little bit fast.
अच्छा ऐसा करें ये तो डेमो वीडियो सर आगे फिर बैठे सर वैसे भी आ जाएगा पीछे जाएंगे आ जाएगा सर आओ चलिए वॉलीबॉल भी चलिए भाई और कोई है क्या बाहर
जो भी हाल के बाहर भी अगर कोई है वो भी प्लीज आ जाए.
हेलो
चलिए हमने ठीक किया।
अजय डॉक्टर साहब अजय
आप बैठ जाए
आप अपने साथियों के साथ हमारे साथ हो। अपने साथियों के साथ कोई ये नहीं है।
थैंक यू सर, थैंक यू बेटा।
मैं कुछ तो लिख देंगे।
नहीं हम किस तरह
एक सेकंड
आप भी आइए।
Thank you. Hello. Now we can have the tea. Session will start